This is going to be the laboratory associated with the anatomy of cells and also cell division, so mitosis. So I want to begin with just looking at a model of a cell. And so this is one of the models we have in the lab. So if we look at the different parts that we've discussed so far in the lecture, we have a cell membrane, we have a nucleus, one nucleus in this case, and then we have all the organelles. So as far as kind of starting with the nucleus, we got the nucleus. You can see it's also a double membrane, so lipid bilayer. There's nuclear pores to allow things in and out of the nucleus. And then there's a dark spot in the nucleus called the nucleolus. Some of the key organelles that we're responsible for include the rough endoplasmic reticulum, which is here. And you can see the white dots represent the ribosomes on it. This kind of maroon structure right here would be the Golgi apparatus. So remember, the proteins that are made on the rough ER go to the Golgi apparatus to put the finishing touches on them. These double-walled organelles right here are called mitochondria, very important for ATP production. And then the other key structures that we've recognized, we see some vesicles in here. These could be lysosomes or peroxisomes, so you can't really tell the difference just by the model. But we also see a centrosome right here. Remember, a centrosome is two parallel centrioles, and it's very important in forming the mitotic spindle. So these yellow tubes right here would actually be microtubules that can expand and extend and form the mitotic spindle. This is a model of a cell membrane, so lipid bilayer. So you're seeing two layers of phospholipids, and the silver structures right here represent the fatty acid tails. The styrofoam balls represent the phosphate heads. The yellow structure right here represents cholesterol. On the surface, we have some carbohydrates. This would be glycoproteins or glycolipids, depending on what they're attached to. And another view here, you can see the same structures, but also you can see an integral protein right here that's actually serving as a channel. Then there's also a peripheral protein right here. So integral protein here also going all the way across. It looks like another one here. And then a peripheral protein just on one side. Here's another model of the lipid bilayer. You see the phosphate heads, fatty acid tails, and the heads orient themselves towards the fluid. The tails migrate away from the fluid, so you get the formation of this lipid bilayer. Going into mitosis, so the different phases looking at models. So we'd have prophase, metaphase. Then the next several models represent different phases of anaphase. This looks like really early anaphase. This is anaphase kind of midway. And this would be more late anaphase, when the chromosomes are almost, or the sister chromatids are almost to the end of the cell. And you see the mitotic spindle formed right here. When we start to get the formation of two new cells, it's telophase, and then the official splitting of the cytoplasm would be cytokinesis. And then looking through the microscope at these phases, one of the lab activities that we do is look at fish mitosis, where these cells divide really quickly. So if you take a snapshot at any point in time, you see a bunch of different phases of mitosis. So some of them are labeled right here. So you can see metaphase right here. The chromosomes going across the middle of the cell. Here's telophase, where you see the two separate cells forming. Kind of looks like two cells that are fused together. Prophase is probably what most of these cells are going to be in. Just looks like a normal cell. Interphase, very similar, just a little bit lighter. I am not going to have you determine the difference between prophase and interphase through a microscope because they look really similar. But prophase, the chromosomes are just darker because they're more condensed. We come to this slide, we see anaphase right here. See the chromosomes or sister chromatids migrating to different sides of the cell. And if we come over here, here's a really good anaphase. So you see the sister chromatids moving to the opposite sides of the cell. And then lastly, just to give you another couple of views, here's definitely a metaphase right here with the chromosomes lining up in the middle. And then you can also see, and this one's a little tricky because you can't see the whole thing, but this looks to me like anaphase, where you can see these chromosomes going that way and these going this way. But if I could be off there, if this is not what I think it is, that could be metaphase. So with metaphase, you're just looking for that straight line going across.